Thank you, Akil, for the introduction. I will have to tell you at the start of this talk, there are quite a number of disclosures that I need to share with you. First of all, I am not a pediatrician. I am not an obstetrician. I'm not a neonatologist. What I am is a pulmonary and critical care physician scientist. I'm an elected member of the National Academy of Medicine. As you can see here, I'm also the CEO of this biotech company called Aqualung Therapeutics. And I'm the director of this center at the University of Florida, which is a Center for Inflammation Science and Systems Medicine. Now, a number of you in the audience are, in fact, physician scientists. You know the goal for any physician scientist is to find something in their work, in their laboratory, in their investigation that can perhaps improve the human health and reduce human suffering. And the goal of the work that I've been doing in my career, as well as in the company and the center that I direct, we're looking for novel therapeutics and the novel inflammation-based therapeutics. And so in the work and collaboration that I've had with a number of individuals, including Akil and including other folks that I'll mention during the course of this talk, we've focused our therapeutic targeting and discovery on trying to address what is the thematic underpinning for this conference, which is the rising infant mortality, and address the absolutely unmet need for novel therapeutics there. You already know all the information that I'll share with the next few slides. This is a slide of the regional variation in the proportion of preterm births that result in neonatal death in the year 2020. And you can see there's quite a lot of heterogeneity in the proportion of preterm deaths. And that includes the 3 to 4% that you see in some regions of the world, but in other regions of the world, the percentage is elevated from 6 to 11%. And here are the top countries in the number of preterm birth rates, as well as the number of preterm birth numbers. Now, the rising maternal and infant mortality is well recognized, as this audience knows better than anyone. And what's already been alluded to earlier is the fact that there is great disparity between the death rates in extremely premature babies born, where in low-income countries, that's 90% that die within days of birth, whereas that's only less than 10% in high-income areas. 13.4 million preterm births in 2020, 1.1 to 2 million neonatal deaths per year are complications of preterm births. And as Akil and several other speakers, including Dr. Carlo, noted, that this is the major cause of mortality in children under 5 years of age. What we are trying to address in the work that we do is to address this global unmet need for drugs that might be able to address some of the issues around preterm births, around maternal and infant mortality. There are no approved therapies that really address maternal and neonatal morbidity or mortality. Recently, the FDA and the NIH had a symposium where they were discussing the need of the unmet needs around maternal and neonatal morbidity and mortality. These were the drugs that were touted as being FDA approved specifically for obstetrical indications. And what you can see quite clearly is that a number of these are primarily for obstetrical indications, and the drugs that are used to target myometrial activity to inhibit uterine contractions and hoping of delay preterm births, they have not reduced the number of preterm births. The drug that was touted as being able to do just that, Makena, a drug to reduce preterm birth risk, was withdrawn from the FDA because of lack of efficacy. So the thematic underpinning for what our work has focused on is targeting immuno and inflammatory responses in hopes that we might be able to reduce the severity of preterm births and maternal infant mortality. The immune responses of the lower reproductive tract are very, very complex. But preterm births are related to intraamniotic infection, a process that, as you know, is chorioamnionitis, as well as intraamniotic inflammation, which is induced by molecules which are called damage-associated molecular pattern proteins, or DAMPs. These are molecules that activate the innate immune system and induce this sterile intraamniotic inflammation. We believe in growing abundance of literature supports the idea that this maternal and fetal immune responses are biological drivers of preterm births. And the mechanisms by which it does that is the inflammation causes edema in the amniotic membranes, causing separation from the uterus, which is a signal for inducing labor and ultimately the births. Antibiotic therapy, as you folks know, has actually improved outcomes and the survival of extremely preterm births, but has not really prevented the morbidities or reduced the premature birth incidence. So they're, again, underscoring the idea that there are no approved therapies to address amniotic infection or sterile inflammation. Now, when I was chief of pulmonary in critical care medicine at Johns Hopkins, my lab identified a protein. It's called eNAMPT. We identified this protein using a variety of genomic and genetic and biochemical approaches. And what we have found is that this protein is an extremely important modulator of innate immunity. And it does this by binding a receptor that regulates pathogen responses in people, which is a receptor called toll-like receptor 4. What we showed subsequently is that the binding of this protein eNAMPT to toll-like receptor 4 is a really critical determinant of the level and severity of a number of disorders, a number of diseases characterized by inflammation and by fibrosis and actually even neoplasia. And these involve multiple organs, including the placenta. And what you can see at the bottom of this slide is that the induction of infection through ascension of bacteria through the cervix into the amniotic fluids, or the induction of inflammatory responses within the placenta leads to a release of NAMPT. And in doing so, NAMPT can accelerate the inflammatory response in these areas that lead to premature births and the complications of prematurity, as I'll show you, and ultimately an increase in maternal and neonatal mortality. Well, we've developed an antibody, a monoclonal antibody. It's called ALT-100. This is a neutralizing antibody that binds to NAMPT. And in neutralizing NAMPT, it reduces the severity of inflammation. We've shown that this reduces the severity of fibrotic diseases in a number of different organs. And we postulated, in collaboration with our colleagues in neonatology and obstetrics, we postulated that this antibody may have efficacy in reducing the untoward effects of prematurity. So when we first found this, identified this protein, like I said, we were using genetic and genomic approaches. There were no papers in the literature around this as a potential lung inflammatory protein, but there were papers in the obstetrics and gynecological literature that showed that this was a protein that was present in amniotic membranes. Now, the protein is normally a constituent of normal cells. Inside a normal cell, this protein is an enzyme that regulates the production of a very important metabolic protein, NAD. But how it functions, and we think, in chorio and sterile intraamniotic inflammation, is that we think this protein works as a DAMP. which means it binds to a toll-like receptor 4 to induce inflammatory responses, and it does so by binding these receptors, as I've already mentioned, and I won't go into much detail there. But this is a very well recognized and a very, very highly targeted mechanism of how you might modulate inflammatory disorders. So the company that I founded, Aqua Lung Therapeutics, we have very strong indications for this antibody to be effective in a number of inflammatory and fibrotic conditions. They're shown here on the left, and we have clinical trials that have been initiated or anticipated to be starting soon. But I want to bring your attention to the fact that we recently have really focused on two disorders really relevant to this audience, which is the potential utility of this monoclonal antibody to reduce the severity of neonatal respiratory distress syndrome, as well as preterm complications of preterm births and chorioamnionitis. Those trials are called SARDINA and eNAMPT. And so let me just tell you a little bit about the antibody itself. This is a humanized monoclonal antibody, a very high affinity antibody for its target, which is eNAMPT. We've manufactured the antibody, and we found that in our manufacturing of the antibody, it manufactures at a very, very high level of expression, which means the cost of the antibody is actually very low when you consider the fact that the antibody lasts in the circulation for 3 weeks. You only have to give one dose to the mother that's experiencing chorioamnionitis, and therefore the cost of goods is extremely low at $30 a dose. We tested this antibody in healthy human volunteers, found the drug to be extraordinarily safe, had no serious adverse events, and it was exactly comparable to placebo. And we next went to test this. Again, I told you I'm an adult pulmonary critical care physician scientist, a disorder that I have been very much desiring to move the needle on the severity of disease is a disease called ARDS, which you are familiar with, and we have a trial called the PORTA trial, which is focused on this particular disorder. ARDS, everyone here knows what acute respiratory distress syndrome is, I'm sure, because this is what patients that had COVID with respiratory illness that went on to the ICU and needed mechanical ventilation, this is what they die of. There's 2 million people across the globe that have ARDS every year. This is pre-COVID numbers. And the mortality rate for patients with adult respiratory distress syndrome is 25 to 40%. The disease is similar to neonates in that they have x-rays that look at what you're seeing there, diffuse alveolar infiltrates, severe hypoxemia, and often primarily require the initiation of mechanical ventilation. The lungs fill up with fluid because of the inflammatory response. This is an enormously inflammatory injury, and the systemic inflammation that ensues causes multi-organ failure, which is the cause of the increased mortality. Sadly, since the last 60 years, we've known about ARDS for 60 years, and there's still no approved therapies for ARDS. So we started this trial targeting the utility of this antibody, the ALT-100 antibody, in a trial called PORTA. This was a moderate to severe ARDS. The drug is given at the onset of the illness, and I said it's only given once intravenously. And then we've analyzed 15 patients that have received the drug to date, and they were recruited at the places that you see in on the map there. And what we have found is really quite remarkable initiation of clinical efficacy, evidence of clinical efficacy. I'll give you two examples because it's not exactly the topic for today, but two examples are the number of ventilator-free days. This is a primary efficacy endpoint in these types of trials. The number of ventilator-free days was markedly increased in the patients that received the monoclonal antibody. In addition, we also demonstrated that the decline in cytokines in the blood was markedly accelerated in patients that had received the intravenous dose of the antibody. So the antibody has proved safe in these very, very sick individuals. It proved effective in these individuals, given that, granted that it's only 15 patients, but we're embarking on the next study will be to extend the number of patients and show that this drug has efficacy in this particular disorder. This leads us to the initiation of a trial in neonatal respiratory distress syndrome, the SARDINA trial, as I mentioned. For these studies, we've done this in collaboration with a close colleague and collaborator, Mohammed Ahmed, who's the director of the NICU, the neonatal ICU at the University of Florida. Mohammed and I have collaborated for a number of years on a number of important projects in pulmonary hypertension, et cetera. But what we want to do starting next year is to conduct this SARDINA trial, which is a strategy to attenuate respiratory distress in neonates with the ALT-100 antibody. Neonatal respiratory distress syndrome, for those of you that may not know this, occurs within hours of birth, and it's a leading cause of morbidity and mortality in premature born infants worldwide. It's the most frequent cause of death in the 1 million deaths that occur in neonates that are less than 37 weeks of age, and again, it's a worldwide issue. The development of respiratory distress syndrome, as we know from Dr. Carlo's earlier talk today, it's related to immature lungs not being able to make surfactant, and so the immature lungs do not function well. They become hypoxemic. They're inflamed. The initiation of surfactant replacement is extremely valuable and reduces the morbidity and mortality in the disorder, but not all babies, especially extremely preterm babies, respond to the surfactant, and the inflammatory response continues to persist in these babies, especially the extremely preterm babies. So again, the innate immunity activation is part of that, and it's also exacerbated by babies that wind up on ventilators. The ventilator itself is a very potent stimulus for inflammation, and these babies are on ventilators for quite some time, and the ventilator and hypoxemia together contribute to the long-term complications the babies endure, which is the development of bronchopulmonary dysplasia, pulmonary hypertension, the neuro developments and cognitive developments that ensue. So reasons that we believe that this antibody will be effective in the neonatal respiratory distress syndromes come from several observations. One of them is that we have very strong evidence in our preclinical models that the antibody blocks ventilator-induced lung injury and also inflammation-induced lung injury. And this is from a porcine ARDS model, in which you can see in the bottom two panels that the animals that received our antibody have a marked attenuation of inflammatory injury. And we also shared with you already that in the humans with ARDS, with inflammatory lung injury and on the ventilator, that the antibody was remarkable in reducing the requirement for ventilation. A second observation is that these two manuscripts that we've published, using again preclinical models, we know that the target for the antibody is increased in the lung tissues of the neonates that are born to mothers that have chorioamnionitis, and that the cytokine expression is also markedly increased in the blood and the lung of these neonates. And when the mothers are treated with the antibody to eNAMPT, the ALT-100 antibody, NAMPT levels and cytokine inflammatory levels are reduced. We also have examined the development of bronchopulmonary dysplasia, pulmonary hypertension in the neonates that are born prematurely, to neonates that are born to choreo mothers. And again, the antibody was markedly effective in reducing the severity of disease in these neonates that have born to choreo mothers that are developing bronchopulmonary dysplasia and pulmonary hypertension. And then finally, in a translational study in collaboration with colleagues at San Antonio, we observed that if you examine the level of NAMPT expression in neonates that are extremely low birth weights, that the NAMPT expression in the blood actually predicts the development of bronchopulmonary dysplasia. So the study that we're doing, Sardina, is an investigator-initiated trial. I mentioned Dr. Ahmed and Aqualung Therapeutics are embarking on this phase 1 trial. 18 premature infants with respiratory distress syndrome will be randomized into 3 groups. The 3 groups will be randomized to placebo or 2 different doses of the monoclonal antibody, and will be identifying the responses to that treatment in a variety of not only the safety issues, of course, not only the pharmacokinetics, but also the effects on neonatal morbidity, days on oxygen, the incidence of bronchopulmonary dysplasia, necrotizing enterocolitis, sepsis, etc. So we're very excited about embarking on this particular trial as an adjunct. Of course, they will receive their standard of care, which is surfactant replacement and antibiotics, etc. But we believe that this antibody has a potential for augmenting the improved survival and to reduce the severity of the complications of prematurity. The other study I'll just want to talk to you about briefly is it's called IMPACTA. This is again, it's impacting the prematurity associated complications with the therapeutic ALT-100 antibody. As I shared with you on the RDS study, the observations that make us think that this antibody may have efficacy in women with choreo that are going to be delivering premature infants is that we've assessed in a number of women the expression of the protein that's the target for this antibody. We assessed their placentas of women with choreo and compared them to normal placentas, and you can see here on the bottom right panel that the expression of the NAMPT was markedly increased compared to normal placentas. We've also measured it's a biomarker, so it's very easy to measure eNAMPT levels in the blood. eNAMPT levels are increased in women with choreo, but they're very close to the levels that we see in just pregnant women, which have an increase as well. But there's a really interesting study that was reported a few years ago, measured eNAMPT levels in the amniotic fluid of women with amniotic infection, with sterile amniotic inflammation, and compared them to controls. And what you see here is there was a 5- to 7-fold increase in the levels of eNAMPT in the amniotic fluids of these individuals. Again, consistent with the idea that this is a very, very viable target for therapeutic intervention. Another observation that was derived from our preclinical studies is that the antibody reduces uterine inflammation in choreo-exposed either sheep, guinea pigs, or mice. Here I'm showing you the fact that the antibody reduced plasma cytokine levels and uterine inflammatory markers as well. And when we looked at gene expression in the uterus of the mothers that received the antibody, you could see that the antibody reduced the expression of highly inflammatory cytokines in these preclinical models. The antibody also delayed the onset of premature births, as well as improved the survival of the offspring in these preclinical models of prematurity. These are in mice on the upper panel, and they're in guinea pigs on the lower panel. And the point is that the delivery of the antibody to the mother with choreo caused a delay in the premature birth and caused a significant increase in the survival of the offspring. And if we examine the lung inflammatory component in the neonates that are born to choreo mothers, there is a marked reduction in the level of inflammation in the lung, as well as in the cytokine levels in the offspring as well. And then finally, the animals in a model of necrotizing enterocolitis, a rat model of necrotizing enterocolitis, the antibody was really quite dramatically effective in reducing the severity of enterocolitis. You can see in the middle panel of the micrograph there that this model of necrotizing enterocolitis is quite dramatic and was nearly completely abolished in rats receiving the monoclonal antibody. So the IMPACT study is done in collaboration. We're conducting the study in Doha, Qatar. Dr. Ashraf God is our collaborator. Ashraf runs the NICU at the largest women's wellness and research center in Doha. They have 18,000 births per year there, and Ashraf tells me that the incidence of choreo in that hospital is around 3.1%. So I think in the first bullet there, which I didn't read, which I'm sure a number of you know, that premature birth incidence and mortality in the Middle East is very high, particularly in the Kingdom, Iran, and Iraq. And we think the IMPACT study, which is a phase 1B trial of the antibody in pregnant women, may actually hopefully reduce the maternal morbidity and improve the complications of premature births. And so this study involves, again, similar to the respiratory distress syndrome in neonates, 18 pregnant women with suspected intraamniotic inflammation and choreo. They'll be randomized 2-to-1 to receive 1 milligram per kilogram dose of the antibody. The exclusion criteria are shown there. And what we'll read out is obviously assess the maternal morbidity as well as the neonatal outcomes, the time to delivery, the development of bronchopulmonary dysplasia, necrotizing enterocolitis, sepsis, etc., as well as an analyze for inflammatory biomarkers. So just to finish up, these are 2 studies that'll be starting in the first part of next year, and the Sardina and the IMPACT studies to utilize this antibody that we've derived in targeting a component that we think is a very important contributor to maternal and neonatal inflammation and severity of disease. And it has the potential, we believe, to reduce inflammation-related maternal morbidity by reducing the level of systemic inflammation and uterine inflammation as well. We think it has a potential to reduce the inflammation-driven premature births as well as the incidence and severity of respiratory distress syndrome, and to improve our neonatal ICU outcomes and survival. And then lastly, it has the potential to really transform patient care. If the antibody is successful, as we hope it will be, it will result in fewer NICU admissions, shorter NICU stays. It will reduce healthcare costs, obviously, if both of those things are accomplished. But more importantly, it will reduce the long-term effects of neonatal premature births with reduced bronchopulmonary dysplasia, necrotizing enterocolitis, and neurologic injuries as well. I've mentioned already that the cost of goods is very low. Wish we could be lower, and we'll work on ways of getting it even lower than that. But at a dose and cost like this, there's a potential for some global impact. And with that, I'll just say, tak su miket. So thank you very much.